Hello and welcome to the Agena Corporate Workshop for PGX. Over the past several years, there's been a rapid increase in interest in PGX. But one of the challenges with pharmacogenomics is what are the right genes and variants to test for? You ask 10 different scientists, you'll get 10 different answers on what are the right panels. So we're pleased and honored to have two key opinion leaders and two thought leaders in PGX speaking today in our workshop, which is centered around the guideline-driven PGX testing. Now, she really needs no introduction for those of us in the PGX space, but Dr. Vicki Pratt is currently the Director of Scientific Affairs for Pharmacogenetics at Agena Bioscience. She, of course, is the past president of this association for AMP, and she's been a part of several consortiums, roundtables, real thought leader in the space. We also have joining us, speaking is Dr. Chad Bozeman, who's Associate Professor at the Department of Medical Genetics at the University of Calgary. Chad also is part of many organizations, of course, including CPIC, PharmVar, PGRN, Stripe, and of course, in all of his spare time, he's also the CEO of a company, Sequence to Script, for generating reports and interpreting PGX information. So without further ado, I am very pleased to turn it over to Dr. Vicki Pratt.
Hello, and thank you everybody for joining us today. I'll put my glasses on so I think I can see what I'm talking about. So we're going to talk about guideline-driven PGX testing and our new targeted testing approach for covering AMP Tier 1 and Tier 2 PGX recommendations. So in PGX, precision medicine, getting the right dose to the right patient at the right time with the right test. So there's a lot of tests out there, and all these tests include different genes. As Glenn indicated, you say, what gene should be on there. They're going to put something on there. Chad and I's favorite test. If you want to know what a crappy test is, don't order one with MTHFR. That will be a crappy test. What genes should be included in a clinical test? As duties and past president and in charge of the AMP PGX working group, we came up with a best practice statement regarding pharmacogenetic testing. And this came about largely in response to the ANOVA letter that the FDA had sent to a lab that was doing pharmacogenetic testing. But from AMP, all health-related pharmacogenetic claims must have well-established clinical validity. They must comply with CLIA. You must have a report that's understandable by healthcare providers. If you just put star 1, star 2 for 2D6 or 2C19, I can tell you most healthcare providers and most physicians, nurses, nurse practitioners have no clue what that means. So you have to have something that's understandable by the healthcare community. And then patients should not change their treatment plan based on any pharmacogenetic result. So what guidelines are there? Well, there's the FDA drug label, the Clinical Pharmacogenomics Implementation Consortium, which I'm a part of, the Dutch Pharmacogenetic Working Group, the Pharmacogenetics Knowledge Base, or PharmGKB. There's many out there that can provide some sort of guidance around pharmacogenetics testing or how to interpret those results. This is a screenshot from quite a while ago, and it's now much larger, of what some of the guidelines that are available on the CPIC website. So in CPIC, for those of you who may not be familiar with it, but I can see there's many PGXers in the audience here who've seen this before, is that based on in the CPIC guideline, the CPIC doesn't tell somebody when to test or not test. CPIC says if you have a PGX result, this is how to interpret those results. So if you're a star 1, star 17 with clopidogrel, you get the normal dose of clopidogrel. If you're a star 1, star 2, like I am, don't take clopidogrel. It won't work for you. So just sort of an example. So The earlier example that I had put with some of the laboratories that had lots of genes and different genes on there, there was one lab that actually had BDNF. I'm like, I don't even know what BDNF is. So I went to the PharmGKB website and looked at it, and if I look at the evidence around it, according to PharmGKB, it's sort of level 3 evidence. It doesn't have great evidence for some of the drugs listed up there. So is really that a research gene? Why are we doing research on clinical tests? Doesn't make sense to me. So this has been my passion project for the past 8-9 years, is really what gene or what variant should be included when you're testing for PGX genes. If you look at, this is from one of my papers, and if you look at different testing platforms, this is probably an older one, you can look across and all these labs or all these platforms have different variants that they include in there. Which ones are relevant, which ones are a normal functional allele, which ones are decreased functional allele. It just doesn't really make sense what's being included in some of these. So this is my passion project, standardizing the variants that are making recommendations for the variants that should be included in clinical testing. So this is my team. I see at least one in the audience. If you haven't seen the papers, Karen Weck is also my co-chair and partner in crime. But we have people from not only the US, we have people from Europe on the team. We usually always have a junior member to get them involved and get them passionate about writing guidelines and recommendations. So one of the things we had to come up was a framework for for how we were going to review what variants should be included in clinical testing. Again, many of you are familiar with PGX, but most all of the genes are reported using the star allele system that represents haplotypes rather than an individual variant. And they're typically named in the order of discovery. So, you know, you can have a star 2 and a star 4, but it'll be different parts of the gene. So they're just named in order of discovery. A star 1 is no variants identified. Nat2 used to be the exception for that, but that has now been standardized in PharmVar, and so now a star 1 is no variant detected. For most genes, an individual will have at least 2 alleles with one copy of each gene, and that's reported as a diplotype, such as a star 1, star 2. And then some individuals may have gene deletions, duplications, multiplications that can involve one or multiple alleles. The diplotype is used to predict the phenotype or the metabolizer status. And CPIC has standardized these terms to ultra-rapid, rapid, normal, intermediate, and poor metabolizer. If you looked in some of the old PGX literature, extensive metabolizer is a normal metabolizer. So if no variants are detected, it's either because they're not present in the test or they're not included in the assay design. Or they're not present or they're not included in the assay design. And the diplotype then is reported as star 1, and reported as a normal metabolizer. So the goal of my working group, or the AMP-PGX working group, is really to standardize PGX testing across all clinical laboratories. Like my podium's vibrating. Inconsistent interpretation can lead to discordant therapeutic recommendations. There's been papers like, this test doesn't match the result of this test, and it's the same person. This is why PGX is crap. But it's part of the design of the assays. And then publications show lack of consistency in alleles included in commercial platforms and clinical tests. So in 2017, the Genomic Medicine Research, or the Genomic Medicine 10 conference, really that was held by NHGRI, really called for assay standardization. And AMP then took up our sort of flag of wanting to help create that sort of standardization. So how do we reduce the variability? Well, we can do sequencing. We can test all known alleles, suballeles. So right now, just looking at 2D6, not including all the duplications, multiplications, there's over 175, not including the suballeles. We can sequence it and instead of doing targeting genotyping, and that's likely coming in the future. But the current state in pharmacogenetics is they're technically challenging by short read technologies. So you got to do a pre-PCR before you do your NGS. And then there's pseudogenes, homologous gene families, duplications, deletions, deeply intronic variants. All of that you can't really do by an exome or an exome background. And then there's challenges in reporting the genotypes. So say you have a 2D6 star 2 star 4, but then you also see a rare variant. The guidelines on interpreting rare variants are for inherited diseases, rare alleles, and it doesn't work for PGX because PGX is not rare. Everybody has I will guarantee you in this room, every one of you has a variant in a PGX gene that affects your metabolism. And then there's really challenges in interpreting those rare variants and alleles. So we decided to go along and do something similar that we did with ACMG in our recommendations for cystic fibrosis, is define a minimum set of variants based on multi-ethnic allele frequency in order to optimize the diagnostic test rate. I didn't want to test all white people variants. That doesn't solve anything for PGX. So we came up with this sort of expert opinion consensus recommendations. Our tier 1 or our must-test alleles have to be well characterized, effect on function or protein expression or gene expression. They have to have an appreciable minor allele frequency in a patient population. So, and why I don't say a cutoff, because some of the variants are more common in some genes than other genes. Some of them they're much more rare. So each gene we look at individually, we look at the allele frequencies and we make a cutoff. There has to be available reference materials. This is to ensure quality in laboratory testing. ACMG came up with their CF100 list. Of those CF100, almost half do not have a reference material, so you don't know whether there's quality in testing or you can get a false positive or a false negative. It just doesn't make sense to me. They didn't ask me, but I could have told them what I thought. And then the technical feasibility to detect variants in a clinical laboratory, we added that requirement for 2D6 because some of the variants in 2D6, the hybrids and some of the things are really very difficult. And so there's a VNTR in T15. Some of these variants are really difficult to detect either on standard assays and even NGS assays. So we would downgrade some of the variants based on their technical difficulty. So knowing that, just giving you the bare minimum, everybody's going to say, my test is better because I do more than the bare minimum. Okay, I lived through cystic fibrosis. So we created what we thought was an extended panel. So it met at least one, but maybe not all of the criteria to be in tier 1. Maybe the allele is a little bit lower. There may not be a reference material. There's something there that we said, this is still an important variant, but it's less common. There's no reference material. There's something about it. So we had created what we called tier 2, our extended panel. And then we actually commented on other things, and they weren't. Some of them are commented in some of the papers, some of them aren't. Or there were variants with unknown or uncertain function, are usually not recommended for inclusion in any clinical test. But we did comment on some variants. I think it was CYP used to be 3A5 1G. There's a lot of or 3A4 1G. There's a lot of labs that test it out there. It doesn't make sense. You know, we actually said don't test this for this variant. We started off, and if you ever work on any one of my teams, I'm going to keep you on target. 2018, we published our first paper. And I want you to notice how long the titles keep getting through all these papers. So it was only a report by AMP for 2C19. Then a year later, we published 2C9. Okay, now we've added CAP onto our team. And the following year, in 2020, we did warfarin. Since we started with the 2C9, we looked at all the other variants involved with warfarin and warfarin metabolism. We still kept CAP. That one didn't get that much longer. Okay, now we started adding a lot of people in it. 2D6. This one took us a little bit longer, but notice 1 year later, we still published 2D6. Now we've included the Dutch Pharmacogenetic Working Group, as well as ESPT to our team. A year later, we published T15 and TPMT. Again, my titles will keep getting longer. And where are we? 2023, we published 3A4 and 3A5. And then this year, just published in October, we published on DPYD. So I'm let Glenn go in it a little bit more after we're done talking about it, and Chad will talk about it. Here at Agena, we came up with Veridose Core 2.0. It has all AMP Tier 1, Tier 2 variants on it, except for DPYD. There were issues, or the allele frequencies changed between when this was set in the publication, but it has only clinical important variants, all CPIC level A, B genes on it. And so, in conclusion, the Veridose Core 2.0 has CPIC AB level genes, all AMP Tier 1, Tier 2 variants, except for DPYD, and then its clinically relevant panel to enhance research.
Thank you, Vicki. Yep, so I'm here to talk about the Calgary experience with the 2.0 array. So we were one of the first to beta test this array. Before I do that, I just want to give a little bit of an outline. So I'm going to give a bit of background on the Calgary Pharmacogenomics program that we did the beta test in before I get into our evaluation workflow and some of the results and takeaways. So just a bit of a background. Our program was initiated in October of 2020. It originally was just for Alberta, which is just the province over, for those just over the mountains, for those of you that aren't familiar. But then we actually spread it to the whole Western Canada over the following 3 years. Our target population are youth with mental health conditions. So we define youth as 6 to 24. And if they are starting or switching a psychiatric medication, they're eligible for our program. And we are averaging about 500 tests per year, although that continues to grow year over year. If you want more information, I just put the QR code up there because I don't have time to get into our program, and it's not really the purpose of this talk. But if you're curious, scan that or just pull me aside afterwards. So our workflow is physician referral. I guess you do get that vibration. Something's going on underneath us here. So it is a physician referral program. So once we get that referral, we contact the youth or the parent, depending on the age, and we send out a saliva collection kit to their home. We do that through the Canada Post, with some of you probably have heard that they're on strike right now, so that's causing us some grief. But typically it works quite well. They return that kit via the post, and then we do the testing with Alberta Precision Laboratories, which is our provincial public laboratory. We have 2 mass arrays, and then the data from that goes into our clinical decision support software, which is sequence to script, which I'll touch on a bit later. But it produces 2 main outcomes for us. One is a patient wallet card that we send to the youth or the parent, and then the prescribing report, which is forwarded on to that referring physician. And we've also teamed up with our local clinical pharmacology physician consult service, which is a peer-to-peer consult if needed. So when we were looking for a PGX panel, we had a wish list. This is the top 5 things that we wanted. The top of the list was that we wanted it to be evidence-based. So we really wanted to make sure that the gene content that we were including on it was supported by guidelines. So Vicki already talked about the CPIC, Dutch. We also include FDA on there if it was on if it had guidelines within the label. We also wanted the content to be actionable. So those alleles that we're testing, we wanted them to have some effect on the function of the gene or the product of the gene. So we didn't want to do any testing of alleles of uncertain or unknown function, because that just actually irritates the physicians when they get an indeterminate result after waiting for it. We also want to be equitable with our assay, meaning that the alleles that we're testing, as most places around the world these days, it's admixed. So we want to make sure that all the alleles that we are testing were appropriate for the population, and as Vicki said, not just doing a test that's good for white people. And then, of course, efficiency. So because we're focused on mental health, the turnaround time, typically these physicians want it quite quickly. They don't wait too long before they want to prescribe. And so turnaround time is key, and we obviously need an easy workflow, something that we can actually deliver on a quick turnaround time. And then finally, it needed to be compatible with our current clinical decision support, which I mentioned was sequence to script, and we needed to make sure that it could integrate with that system. So first I'm going to show you some like a side-by-side comparison of our current panel and the one that, the 2.0, which we beta test. So our current panel is the Veridose 1.0 with some customization. Mainly we just added NUD T15 and TPMT to that panel, but otherwise it's the off-the-shelf version 1.0 core. When we look at the evidence base, again, this is one of our wish list items. Those in the orange there there's 55% of those genes that were actually in a guideline of some sort. So we have a lot of content on there that we weren't reporting, so we were kind of putting that aside for research purposes. Of those genes that are on this panel, 76% had some type of an actionable allele. So there were some alleles that we could end up with indeterminate results. So that wasn't always great, but they're relatively rare, so it didn't happen very often. And getting to that equity bit, 71% of the tier 1 and tier 2 alleles were covered on the version 1.0. So you can imagine we were pretty excited when we heard that 2.0 was coming out, because that pretty much ticks 3 of our wish list items in that 100% evidence-based, actionable, and equitable. But the bonus here is also that you can see those ones that I've underlined and bolded. We got some additional content along the way as well. So we were particularly interested in DPYD and UGT1A1 because our cancer, even though we're focused on mental health, our oncology group was getting very interested in what we're doing. And you can see in our current panel, we don't have those 2 genes. And then the HLA genes were also quite helpful because in psychiatry, we actually do prescribe drugs that are important to those. Okay, so side to side, it was a pretty much a no-brainer that the 2.0 was a winner, but of course we wanted to take it through our workflow and make sure that it performed well in our hands. So to do that, first we selected 39 samples that we had previously tested on our current panel, that 1.0, and we enriched these 39 for those that had variant alleles, maybe some trickier ones, as well as for CYP2D6 ones that had the copy number variants, so hybrids, deletions, duplications. We really wanted to put it through its paces. So we put those samples onto the 2.0 panel, in collaboration again with the Alberta Precision Laboratories. And then we looked at the between assay comparison, 1.0 versus 2.0. And then to be completely comprehensive, we also wanted to take the outputs from the Agena reporter software and put it through our clinical decision support pipeline to make sure that there weren't going to be any hitches or hiccups with us developing our prescribing reports. And then finally, we wanted to look at lab-to-lab variations. So Agena kindly sent us 20 samples that they had already tested on the 2.0 in their lab in San Diego, sent them to us. We tested those same 20 samples, and so we could compare the Agena versus the Calgary lab results. So here I'm showing you the between assay comparison, and this is the genotype concordance. So you can see it's pretty darn good for most of the genes. You'll see two outliers right off the bat. So CYP2D6, CYP2B6, 50% genotype concordance. So at first blush, that doesn't look good, but I can tell you that that's all because of two alleles, star 4 and star 6, that are on the new 2.0 assay, but not on our 1.0. So that was the reason for the discordance. And then for CYP2D6, we had a little bit lower genotype concordance than you would typically want to see, but those differences can all be explained by just differences in C and V hybrid calls. So in our current one, like we would call star 2, star 4, and with the 2.0, I think the C and V calling seems to be a bit more sensitive and accurate. It actually picks up the hybrid star 68 that we didn't pick up with our 1.0 array. So that explains why we're seeing the lower than expected concordance. When we look at phenotype concordance, though, it's pretty much all above 90%, which is pretty good. From the efficiency standpoint, as I mentioned, that was one of our wish list items. Look, both are pretty efficient. That's why we went with it in the first place. But the 2.0 does give us a little bit more advantage in that we can do larger batches than we did before. So up to 43 samples. Typically, we just do one at a time, so it's 22. It also gives us the setup of only 4-well assay versus a 6-well assay, which our lab techs appreciate. And our lab techs also have told me that the reporter software for 2.0 is slightly more user-friendly than the 1.0. So we've won some thumbs up from the lab group, which is always good. From a turnaround standpoint, it's negligible. It's still pretty fast. Less than a day it takes us to turn a sample around. And then that last bit, we wanted to make sure that it was compatible with our PGX reporting workflow. As I mentioned, we use sequence to script. And so this just ingests the file that comes off of the mass array, out of the reporter software, the Agena reporter software, puts it through that decision support. It marries up all the guidelines, so CPIC, Dutch, and the FDA, and then produces a compiled report in that wallet card. And so we did test that. We used the new report that comes out from the 2.0, put it into our system, and we didn't have any issues with it at all. It actually works quite nicely. And then finally, just the between lab comparison. Not much to say here. It's good, as you'd expect. There was a slight discordant there for CYP2D6, again, owing to a little bit some CMV differences between the labs and some settings, but nothing that is, to me, a major concern. So to close, we're adopting the Veridose 2.0. We're definitely going to make that switch from the 1.0 of the Veridose to the 2.0 for three main reasons. One, it ticks off our two wish list items at the top. It's 100% evidence-based for both the genes and the allele coverage. So everything we test, we can report, which is a huge thing for us. Having to hold back results is lack of efficiency, and it just causes us issues going down the line. It also maintains or maybe potentially even slightly improves our operational efficiency. Like I said, the 4-well versus 6-well setup. And the cost, I didn't really mention this, but because we're using custom assays right now, there's a cost to that, and the order time that it takes. We no longer need custom assays. We can just use the off-the-shelf 2.0, which is really nice. And then finally, it integrates seamlessly with our reporting software, which is a big win for us. We don't have to redo or remake our workflow. It plugs right in, and we're ready to go. So I'll finish there. I'm happy to take any questions. And thank you.
But I just want to add on and talk to the fact that you've already heard about the content and everything. There's been a great discussion on content. And that's exactly the dilemma we face as developers, right? Is like there's not going to be one panel to rule them all. Everyone is going to have their own unique needs. So that's why we developed Core 2.0 with the clinically actionable AMP Tier 1, AMP Tier 2, obviously with the minor discrepancies with DPYD, as Vicky mentioned, because the new guidelines came out as this was in verification with us, but we're rectifying that. And to Chad's point, he went from from 6 wells to 4, and that's exactly what we can do at Agena. Is we have the ability with our custom content ability with our Assays by Agena team. So we have 20 scientists in Agena for exactly that reason, because everyone has unique needs, right? You may have a particular area you're focusing in. It may be mental health, it may be cardiac, it may be pain, so you might need some slightly different genes. So the whole point of Core 2.0 is to act as the backbone, as the foundation, those clinically actionable variants in a smaller number of wells. So that frees up additional wells with the ability to do customization at low cost with high multiplexing. And that's exactly what Chad was doing before, right? With customization, adding additional wells, and many of our customers do. A significant portion of our business is just that, the customization, because we realize there's not going to be one perfect panel for everybody, but we feel we're getting closer with the one that we've developed with Core 2.0. And the last thing that I'll touch on is the fact that obviously there's a need for, we're a system and we have the tests, but then there's the need for the interpretation. So we're agnostic, but we have partner companies that we work with. I see several of you in here, like Ruben. We also have Genexus. We also have Seek to Script, right? So we work with these companies where they've proven that they can take the data that's off of the mass array system and develop clinically relevant reports for physicians. And we're in the process of updating our website, so stay tuned. You'll see additional information on these providers on our website. But in closing, I just want to, again, this was a great discussion. All the points you've mentioned are exactly what we deal with when we're considering developing new products. So love the insight and the feedback and the passion within this group for PGX. So thank you.